Thank you. Well, hello, everyone. Can you hear me? It's great to be here. My first visit to Johns Hopkins, arguably the best and greatest medical school in the world. It's really an honor to be here. Today I'm going to give you, just in sort of different parts of my talk. One part is devoted to sort of, what really gets me excited about being here and having this opportunity to try to figure out how living things work, sort of big picture part. And then I'll try to get a little bit of science in as well, talking about some of the work we're doing on germline and how germline protects itself and maintains its immortality. But living things are so incredible and so complex and amazing that you really can't put it to words. It's hard. It's really hard to convey the incredible complexity and fascinating inner workings of living things. So I found that I can put it to music, though. And so I'm going to show you, and this first slide is actually a movie, and hopefully you can hear it. I have to put this mic down here. All right. Ready? Here we go. It's going to speak for itself.
Take me to the magic of the moment on a glory night where the children of tomorrow dream away in the wind of change.
Those are worms stuck in an air bubble, by the way, in case you didn't know. Little air bubble on the agar plate. They can be a little death trap for the worms. Yeah, so living things are incredible, and that's sort of one of the themes of this presentation. We always forget, I think, how amazing they are, and we always underestimate them. So, when I was in Stockholm with Andy, they were recognizing the work that led to mapping of the background cosmic radiation from the Big Bang. And the really amazing thing about that, they were asking us questions like, well, how is the work in physiology and medicine related to the work in physics? And, it was like, think for a few minutes, and then, well, life itself exists on a cosmic timescale. RNA interference itself is billions of years old. In fact, life on this little planet of ours has been going strong for about a quarter of the history of time. That's amazing. It's a huge amount of time. And germlines, like ours, have at least an opportunity at immortality. Of course, it's limited by the lifetime of the planet. But let's take a look at just another perspective. Thinking about the cosmos, what is our place in it? Where do we come from? Here's an image that you'll all recognize from the night sky of the Big Dipper. This little slice right here is a piece of the night sky where there are no stars in our galaxy. And the scientists who run the Hubble did this amazing experiment. They directed the telescope into this little patch of the night sky where there are no stars in the way. And they just left the shutter open for like 12 days of continuous image capturing. And what they found, they call, I forget what they call it, the most amazing or most important photograph ever taken. You can see this little strip here translates to this. And here's just one little square of that blown up. And every single speck of light that you see in this picture is a galaxy, okay? And we're looking at some of the galaxies here that are really redshifted, really, really far away. They're almost over 13 billion light years away. They're as far away as you can see. And in fact, if you know your cosmology, the story goes that during the first second after the Big Bang, the universe expanded faster than the speed of light. So in fact, the limit of what we can see in the universe is far away from the limit of what's out there. But even just extrapolating from this field of view where every single speck is a galaxy, they can estimate there are about 200 billion galaxies in the visible part of the universe that we can see. And each of those has about 200 billion stars. So if you do the math, it's like more than a mole of stars in the visible part of the galaxy, of the universe. Anyway, imagine what it would be like if you could go there, go to one of these galaxies and find somewhere a star with a planet that has life. What would it be like? Well, you're here. That's where you are. And it's really amazing when you think about the history of life. There's so many amazing episodes in the history of life. It's just really incredible. There's the equivalent of the Big Bang of, for life. We call it the Cambrian explosion. When suddenly, in the history of the Earth, all of the major groups that gave rise to humans and worms suddenly appear in the fossil record. It's mystified everyone from Darwin to current day paleontologists. Why all of a sudden did these life forms burst into the scene and start leaving these fossils suddenly at this time in the history of the Earth? And there are clues now from the geology of this planet that reveal some really interesting things about the history of the planet. And I just want to show you a couple of these things, because a lot of biologists don't know this stuff. It's just, you learned it sometime, but you probably forgot. Anyway, below the Cambrian, there's geological evidence for these glaciation events called snowball Earth events. And there have been a few of these in the history of the Earth that have been so extensive that the ice may have covered the planet all the way to the equator, almost extinguishing photosynthesis on the planet. And I think it's interesting that before the Cambrian explosion, there were two of these events. And I think what this is telling us is that there were really sophisticated creatures living here, but they didn't really have the opportunity to occupy the niches that became available when the planet finally became a more stable environment. But just to put this in perspective, look at the history expanded now to show the entire history of the Earth from its formation to the present day. And I'll just take you through this for a moment. So the Earth forms about 4.5 billion years ago, and it forms from the remnants of supernovas, where all of the molecules heavier than hydrogen were generated basically when the star exploded. So all of you are made out of stardust. Every carbon atom in you came from a star that exploded long ago. And then amazingly, and this is still, even though this is a huge amount of time, it's really hard to believe. Just 700 million years after the Earth cooled sufficiently for there to be oceans and an opportunity for life, we see fossil evidence of the first cells. So molecular evolution had to occur in this 700 million years. That's a long time, but still pretty amazing when you think about going from inorganic molecules or organic molecules to actually life. Now, interesting things happened. If you were looking for that planet that has life, one of the things you'd look for is oxygen, because oxygen is a highly reactive molecule, and therefore when you see oxygen gas, that's a sure giveaway that there's life. So early on, there was no oxygen in the atmosphere at all. Photosynthesis hadn't evolved yet. And then cyanobacteria and other organisms related to them began photosynthesizing. And they figured out this really clever way of splitting water to release the oxygen and to grab some electrons for generating energy through photosynthesis. And as a consequence, they began releasing oxygen into the atmosphere. And this amazing thing happened that I bet most of you never heard of. It's actually called the oxygen catastrophe. They released oxygen, gradually modifying the planet's atmosphere. This highly reactive substance got into the atmosphere, and the geologists think what it did is it reacted with methane, which is the greenhouse gas that was keeping the Earth warm, and induced a global snowball event that was 300 million years in duration. That's this blue band right here. That snowball almost wiped out the cyanobacteria, and with them us, right? I mean, because there was no photosynthesis going on, probably very little for quite a while. Amazing episode in the history of the Earth. And then gradually, oceans absorb the oxygen. You start to get a lot of oxygen released. You need oxygen to get big, so this could help explain the Cambrian explosion occurring at this point, because you have aerobic metabolism now. You have an opportunity to make bigger organisms. And whatever the reason, I think it's really amazing to look at this history of this planet. The innovations that were happening inside these cells during this time are amazing. All the information technology that you and I recognize in the laboratory, transcription, translation, all of these big mechanisms, they evolved here. They evolved more probably here. But basically, there's a huge amount of time here. Now, here's the present day up here. Let's just put a couple of more events. Cambrian explosion, if you liken it to the calendar of a year, where the Earth, this is January 1st, the Cambrian explosion occurs on November 18th, so a couple of weeks. This is when the dinosaurs went extinct, okay? That's the amount of distance, 60 million years between you and a little mammal that looked like a shrew, okay? So this is, the point of this is that organisms, all the organisms alive today, they're survivors of all of this evolutionary time. The mechanisms at work inside these cells are mechanisms that function on a cosmic time scale, amazingly complicated. There's a lot we still have to learn about the inner workings of life and how it's able to endure on this kind of a time scale. Okay, so what is the lineage that's immortal? It's the germline. And this is just showing you, an example of this germline of C. elegans. Little writing disappears. You can see the nuclei in the ovary here, and the green stuff are mitochondria. Those are the little endosymbionts that are inside eukaryotic cells that help us metabolize and make energy. And so you see the nucleus, the DNA is in there, and then these little red dots around the surface of the nuclei. You can also see these red dots here. These are germline granules that are full of RNA and protein, often called germline nuage, and they're packed full of the RNAi machinery that I'll be talking about in my talk. And these guys, and one of the things I really like about this, you can just see the information flowing here. We think about the information in the cell, we often think in terms of DNA. There is information there in the DNA, a lot of it, but there's a lot of information in the cytoplasm. There's a lot of information in RNA form, in protein form, even in membrane form. A lot of that information was never encoded in the DNA. Think about it. Where did the original organisms have their information stored? It wasn't in DNA. It was in RNA and various other types of places inside the cell. So we have a lot to learn still about where the information is, how it's stored, and how it's being managed inside the cell. And this image just really shows this information flowing to fill the egg. And you can see these granules that are full of RNA that are going to program gene expression in those resulting progeny. So here's the real Nobel Prize winner. This is that beautiful, elegant little worm, C. elegans, about the size of a comma on the page. You saw its embryos developing in the first slide to the music. And everywhere on this planet, there are little nematode embryos undergoing that same developmental program. And it's beautiful because you can watch it unfold in just a few hours under a microscope, and you can see every cell inside the animal's body. For example, you can see the oocytes inside the ovary. You can see the mouth here. You can look at the neurons. It's just a great model system. And it still has a lot to teach us. One of the things that I put this slide up here is, if you really want to learn something about the basic mechanisms of, let's say, immunity or how gene expression is controlled, study an organism that eats dirt, right? That's where you should look, right? Worms live in the soil and they eat bacteria. This guy lives in the seafloor and filters its food from the mud. And basically, we have a lot to learn from these guys too. These quahogs, the ocean quahog, can live to be over 400 years old. And it's amazing. Certainly people should study long-lived organisms like this to figure out how they do it. How do they manage to keep their body alive for such a long period of time? The germline is even more amazing because it's the immortal lineage. So that lineage can go on forever. One of the ways that the germline is managing that information and allowing itself to be protected, for example, from invading organisms is shown here. This protein crystal structure is the structure of the Argonaut protein bound to a guide RNA in red and a target portion of a target RNA in blue. And this mechanism that you're seeing is the equivalent of the Google of the cell. This is a search engine for the cell that allows the cell to use a short guide sequence to find and regulate information after it's already been expressed. And again, we always think about things happening, at least I do, inside the cell in sort of an anthropomorphic way. But it's not like that. Inside the cell, for example, transcription is occurring at the rate of 50 nucleotides or so per second. And translation occurs at a rate of about 20 residues per second. So these machines that transfer the information from the genetic code into protein, for example, are incredibly fast. And they're working in this sort of space where molecules are colliding at the rate of millions of collisions per second. And they're sorting through this and finding the right information to put these pieces together and to express a gene accurately. And organisms have been in the information age for 3.8 billion years. And only now, I think, now that we're in the information age too, can we begin to appreciate even what that's actually like. What's the first thing you need when you're swimming in that kind of information? You need tools for searching it. And it's going to be, I'm sure every organism is going to have its ways of doing this. One of the solutions is to use this type of mechanism where you use an RNA guide, you use a short piece of sequence information, and you use it to find matching information. This is the basis for RNA interference. It's basically, it's like the Google of the cell. And just this will rotate so you can see how it looks. You can see the double-stranded RNA here, and it's very precise because it's base pairing driven. And you can find, this mechanism can find essentially any gene in the cell because they're sufficient. Even though it's short RNA, it's about 20 nucleotides long, that's sufficiently long to allow it to recognize almost any gene uniquely. And then you have a catalytic center where this blue strand, the target strand, can then be cleaved. Now, one of the things that I really like about Google is that you don't have to be a great speller, and it still finds the thing that you're looking for, right? So cells figured this out too. That's how they use the microRNAs, which probably many of you have heard of or work on. MicroRNAs allow mismatching, and because they allow mismatching, their target repertoires are huge. So if you allow misspellings, of course you'll pull up more web pages, but usually you can find the right one. So that's the sort of I'm feeling lucky feature. And one of the Argonauts I'll talk about today, Peewee Argonauts, actually use the same sort of mismatch pairing to accomplish some pretty amazing things too. So another great thing about the information age that we're in now, that we finally recognize, is that you can, when you work on a model organism like C. elegans, you can rapidly identify genes that are important for the biology of this animal, and then you can go from those genes to the genes that are related to those genes in every other organism for which a genome sequence has been determined. And when we did this first in 1999, we cloned the RDE1 gene. We were, before that, 98, we published that paper. We were really nervous that paper wouldn't get accepted. That paper that was in Nature, with Andy Fire, we were really nervous because it was purely phenomenological. All we knew in that story was that if you give worms double-stranded RNA, they responded to it in this amazing sequence-specific way. So you could even feed the worms double-stranded RNA, and they would mount a sequence-specific response and turn that gene off. And that, as cool as that was, we thought they're going to ask us for the mechanism. I know they're going to, reviewer number 3 always says, yes, it's an interesting study, but there are no mechanistic insights, and therefore, anyway, I won't go into it. But we needed to figure out mechanism, and we didn't know, of course everybody said, well, it's a worm thing, no other organism is going to do this, this is just bizarre. But anyway, we cloned RDE1, and then so we immediately blasted it and looked at all the genes that are related to RDE1. This gene, when you knock it out, completely wipes out the double-stranded RNA response. There's no response at all. The animals are just fine. They grow beautifully in the laboratory. There's nothing wrong with them. They look beautiful, but they don't respond to double-stranded RNA anymore. There's no double-stranded RNA response in these animals at all. But interestingly, and incidentally, the paper describing RD1 was just rejected the day before we cloned it. So we cloned the gene and sent it back to the journal, and it got published. But without cloning the gene, they weren't going to take the paper. So we cloned the gene, and it turned out to be related to this big gene family. So there are 4 human genes over here that are related to RDE1, and you can see by this tree that they're distantly related. And there are 4 more human genes over here that are also distantly related to RDE1. You can see they're way over here. But the thing that was really cool and very exciting for us was that if you look right here, there are worm genes that are more closely related to those human genes than RDE1 is to those worm genes. So these are worm homologs that are very similar to the human genes and not so similar to RDE1. This is a worm homolog that is very similar to these human genes, but not so similar to RDE1. So what we learned from this is that the common ancestor of worms and humans about 1 billion years ago already had 2 argonauts. One that we call the Peewee argonaut group and one that we call the Ago argonaut group. They already had them way back then. And they were already probably doing different things that are important because all animals have 2 members of this argonaut family, the Peewee member and the Ago member. And it turns out that the microRNAs function in the worm in these 2 argonauts and in the human in this argonaut. And another type of RNA called the piRNA functions in this group of argonauts and in this argonaut in the worm. So this is germline anti-transposon defense, recognition of non-self, and this is regulation of your own genes by using a microRNA, which is expressed from the genome like a hairpin RNA, sort of endogenous RNAi mechanism. Okay, and so we were really intrigued by these findings, and we noticed that worms, have like 28 or 25 of these argonauts, and you can see a big group of expanded group in this family of proteins that are all C. elegans genes. And it turns out these are required for the RNAi pathway for silencing, and I'll come back to them later on. This argonaut here is an essential gene that I'll talk about a little bit too. But before I do, I want to just come back to soil and animals that eat dirt, because this, if you haven't already done experiments with CRISPR, you're going to be hearing about it and you're going to be doing it. This is amazing. And again, it comes from little bacteria that live in the dirt, and they get infected with viruses. And I really sort of had to do a double check, because I was telling my 13-year-old daughter that bacteria have this cool antiviral mechanism, and she goes, "Wait, bacteria get sick?" But yes, they get sick. There's like millions of viruses infecting bacteria. But the amazing thing about this system, if you haven't worked with it yet, is that it's a lot like RNAi. It's not Argonaut-mediated mechanism. Rather, it's very analogous, though, in that what happens is you get infected. In this case, it's a DNA virus. The virus gets chopped up into pieces, and pieces are stuck into this chromosomal site called the CRISPR locus. And then later on, the animal can re-express these little RNA fragments. Then incidentally, the size averages around 20 nucleotides, just like the guides for Argonauts. They get loaded onto an enzyme encoded by the CRISPR locus that is a DNA nuclease that's programmed by RNA. So it's very much like the Argonaut, only this is a nuclease that targets genomic DNA. And the amazing thing is that it works incredibly well in everything from plants to animals to you name it. People are doing this now in everything from livestock to crop plants to human cells, and the applications are stunning, astonishing. And it works incredibly well. My lab is using it too, and I've worked in the DNA transformation field my whole career. It's revolutionary. All of you should be designing your CRISPR guides right now. It works. Everybody's getting this to work. It's just amazing. And again, it shows you the power of information tech, right? This is an immunity mechanism in which the animal develops adaptive immunity to an infection by acquiring the information from the foreign sequences, incorporating them and re-expressing them. Very analogous to the way the RNAi system works, except in this system there's actually a DNA copy made inside the bacterial cell. So the worm is doing something even more incredible than these bacteria, even cooler than that. And I can tell you, this is just the only science I'm going to tell you is the next few slides. I'm going to relate to you this model for how the worm is handling this in a really, really amazing, stunning way. So the worm, so the bacteria has to get infected, right? And once it's infected, it can become immune to that sequence by defending itself constantly against that sequence. What if you could remember every gene that you've expressed before in the germline? And then you had a way of keeping that memory and using it to check every new piece of information that comes into the cell. And if you haven't seen it before, you could silence it. That's what the worm is doing with its Pewee system. And I'll just sort of take you through the evidence for that model. So first of all, a key player in this model is RNA-dependent RNA polymerase. Oh, I should show you just in this slide. This again, these are germline cells in the ovary of a nematode, and you're seeing the DNA in the middle, and then this halo of red dots are actually Caesar-1 protein decorating the nuclear periphery. And basically, this protein RNA granule contains not only Caesar-1, but also members of the Wago family. You can see here in green, same structure. And also members of this other Argonaut, the Pewee family, that is also in this same exact structures. These three different Argonauts are doing very different things, but they reside together in the germline in the same location. And indeed, it's interesting, these small structures seem to be docked around the nuclear pore, where they're poised to interact with transcripts that are exiting at the nuclear pore. So here's the model. The model is that Caesar-1 represents a memory of self or active genes in the germline. The Wagos represent a memory of non-self, and the piRNAs are scanning for foreign sequences constantly. And so to just remind you how RDRPs function in the RNAi pathway, And humans don't have RDRP, so it's not clear that humans can quite do it this way, certainly. You have double-stranded RNA entering the cell here in the RNAi pathway. It gets processed by dicer, gets loaded onto RDE1. The strands are separated. RDE1 is then able to cleave its target and recruit RDRP. RDRP then amplifies this silencing signal, which then gets loaded onto the WAGO argonauts. WAGO, and this cycle expands and amplifies the response. This is why worms are able to respond to even a tiny amount of double-stranded RNA in their diet is enough to initiate this cascade, which results in this amplification and leads to silencing. So that's what happens when you give them double-stranded RNA. And back in 1999, when we cloned RDE1, we thought perhaps members of this gene family would be functioning in different types of foreign molecule recognition or nucleic acids recognition, because we knew that in RDE1 mutants there was completely no RNAi left, no double-stranded RNA response left, and yet the transposons were still silenced. So, and yet other RNAi deficient mutants like RDE3 when they were mutated, the transposons started hopping all around in the genome. So we knew that RNAi-related pathways are silencing the transposons, even back in 1999 when we first published the results from this genetic screen. But RDE1 was not required for that. And so we hypothesized that there would be other, perhaps RDE1 homologs that are responsible for this mechanism that's involved in recognizing and silencing transposons. And that's what we think this PIWI system is actually doing. So one of the great things, again, about the information age, right, is that just like at Google where they read your search queries, we can now read the search queries in the cell, just like the NSA is reading all of your search queries. So it's really powerful. You can, for example, pull down the Caesar1 argonaut and ask what small RNAs are in that argonaut, and that can tell you what that argonaut might be functioning, what process it might be functioning in. And the amazing thing about those experiments was they led to the discovery that the Caesar1 argonaut is targeting the animal's own genes, things like ribosomal protein mRNAs or DNA polymerase. I mean, everything expressed in the germline is being targeted by this Caesar1 argonaut, which was totally bizarre because we think of argonauts as silencing factors. And we still don't quite understand how this works, but it's clear that even though Caesar1 is incredibly abundant in the germline, both in the male and the female germline, and yet, obviously, those genes are not being silenced by Caesar1. So what it was doing was still totally a mystery. WAGO, on the other hand, WAGO1 and also other members of the WAGO family are targeting transposons and pseudogenes. And when you knock them out, the transposons and pseudogenes become active. For Caesar1, it was difficult because when you knock it out, the animal is sterile. So we couldn't really figure out what Caesar1 was doing. But it didn't seem to be silencing its targets. At least we knew that. And both of these argonauts localize in the germline in both the sperm and the egg. In fact, they're in the mature gametes. Both the egg and the sperm contain not only the argonauts, but the small RNAs as well. So they're in a place where they could form memories of gene expression that are transmitted from one generation to the next because they're in the gamete. So the handle that we finally got on how this immunity mechanism might be working came from experiments where we introduced foreign sequences into the animal. And for many, many years, scientists working with worms, including Andy and I, had a really hard time getting DNA from a foreign source to be expressed in the worm. It was really vexing because in the germline, those genes, no matter what they are, if they're foreign, the worm would seemingly shut them down. And it was really hard to get GFP expressed. You could eventually get it, but a lot of time, you'd just be frustrated. Eventually you can get them. But then finally, another lab, Eric Jorgensen's lab, came up with a way of introducing sequences by homologous recombination into a defined location in the worm. And we were able to put the same transgene in over and over again at exactly the same place. For the first time, we could actually try to understand why, if one time it's expressed and another time it's silent, what's the difference? Because they're at the same position, so we don't have to worry about the position affecting how the gene is expressed. So Masaki in the lab actually did this experiment that began when he pulled strains out of the trash can, literally pulled them out of the trash. That's where you should be looking for your next breakthrough. Literally what you do when you try to make a transgenic animal is you spend all this time, you make your construct, you inject it, you get a line, and then you look, is it expressed or not? If it's expressed, yay. If it's not, you throw it away, right? But we work on silencing. And he was getting a lot of these lines that were not expressing, but they seemed perfectly fine, but they weren't expressing. So he pulled them out, and he did an incredibly simple experiment. You can just see here, this is what it should look like, beautiful green GFP expression in the germline, or nothing at all. The worm's here, but you can't see anything. So, and the thing that was so vexing about this, these silenced lines, if that's what they were, they're 100% stable. They never turn on. You can even mutagenize the worms and screen millions of genomes. And we know now you can find the mutants, but it's really hard for a variety of reasons. But he did this amazingly simple experiment that I'll show you in just a second. Just a little more phenomenology, because I think it's kind of intriguing. If the GFP tag is put on the foreign part is put on the 5' end, it's much more prone to being silent. You can see here that this construct is silent 100% of the time. In fact, I told my graduate student if she can get this transgene to express and stay on, then she can graduate. She's still working on it. If you put the GFP over here, it's always on. It can be off too, but in this experiment, it was always off, always on. So there's something weird about the position of the foreign sequence. So here's what Masaki did. Incredible experiment. This is the trash can. This is the great trash can experiment because it takes like 3 days to do. He took a line that was silent. He crossed it to a line that was active. This is what you'd expect from Mendelian segregation. You'd expect a quarter and a quarter in the hets. This is what he found. So line A that is silent could dominantly silence line B. Not only that, after maintaining them together in the first generation, you could separate them again, and line B would stay off. So it would go from a stably on state to a stably off state. Amazing. And of course, we knew right away something really interesting was going on, and I have only 10 minutes, so I'm going to have to go fast through the next few slides. But this work was published last July, a year ago, July. What we think is happening is that piwi, which engages thousands of genomically encoded small RNAs, so there's about 200 microRNAs, there are literally probably 30,000 genomically encoded piRNAs in the worm. They're on the order of millions in the human genome. These are genomically encoded, not as hairpins, but as Pol-II products that are processed and loaded onto the Argonaut. And they're so sequence diverse that everyone's been scratching their heads. What could piRNAs be doing? Because they can target everything. There's so many of them. They're so sequence diverse. And in fact, what we think they're doing from experiments that were published again last year is that the piwi is able to use them with degenerate pairing to recognize foreign sequences. And when it does recognize a foreign sequence, one that's not protected by Caesar-1, it induces RDRP recruitment, loads up the WAGO, and sets up this permanent, long-lasting silencing. And piwi, again, is over here on this branch. And as you would predict from that model, if you introduce GFP-CDK into the piwi mutant background, the transgene is on. It's just fine. If you introduce the already silent version of exactly the same GFP-CDK transgene into PRG1 by crossing, it stays off. piwi is only required to initiate, just as that model suggested. So that, again, was all published. So this is how we think the piwi system works. piRNAs scan. They find imperfect targets. In GFP, there are hundreds of piRNAs that could recognize GFP, no problem. Once they do, they initiate this feed-forward silencing, and they permanently turn that gene off. Very elegant, beautiful system. But there's a problem, right? Autoimmunity. How are you going to avoid silencing your own genes? There has to be some way to protect yourself. That's just some examples of targets in GFP where you get a really good seed match with this piRNA. And then if you look, you can see that there's a small RDRP-derived RNA starting right at this G residue that base pairs perfectly down here. I don't have time to go into all of that. But the problem is that there has to be a mechanism that's protecting self-RNA, right? And this is where Caesar-1 comes in. So interestingly, Masaki and me too, whose project it is to get GFP-CDK to stay on, me too noticed that when she was doing a cross with a transgene that we had had around in the lab a really long time, so this is a GFP-containing transgene that had been stably expressed for probably 10 years and cultured for many, many generations, she could cross this line with a new line that was silent. And this is what Masaki observed with the transitive silencing event. And what she observed was the opposite. So here we have an active transgene that can activate a silent allele. If this represents the protective pathway, this gives us an assay for determining what genes are involved in this process. So one idea might be that the GFP sequences have been adopted as self in this strain. And we called that phenomenon RNA-A. And so she did a very simple experiment. This is just showing you that if you cross these two together, GFP-CDK, which is always off, and OMA-GFP, which is an RNA-A allele, this double mutant will show expression of OMA1 in the cytoplasm and the nuclear CDK-GFP, GFP-CDK right here. But as soon as you separate them, it goes silent, and GFP-CDK goes silent again. So it only stays on when OMA-GFP is present. Interestingly, and so I told me too she can't graduate because it went back off. So she's like really determined to get this to stay on, so she kept them together for 30 generations. And then she separated them by crossing. So here they are. And after 30 generations, in fact, there's no longer OMA-GFP in this strain. This is just GFP-CDK by itself, and we all thought that me too would be able to graduate. She just is a third-year student too, so she was really psyched. But unfortunately, by the 10th generation, it starts to go silent. But it stayed on for 10 generations. So just by being propagated together, there's no selection going on here for GFP-CDK1. It's not even functional. Just by being propagated together, it seems that it stabilizes the expression state. And we're looking now at chromatin to try to understand this. Fascinating biology, because you're seeing an adaptation of this allele to be able to sustain its expression by itself. And we knew from our old experiments that if you look at a transgene that was sensitive to RNA-E, what you're seeing here are endogenous siRNAs in the Caesar-1 pathway that target the endogenous part of the transgene right here. The GFP part has very little or no small RNAs targeting it. Even though GFP is being expressed, they're very low signal of Caesar-22 small RNAs targeting the GFP. So the idea was that an old transgene like OMA-GFP would have more, and indeed what we see if we look there, in the OMA1 portion of the transgene has lots of Caesar-1 small RNAs, and the GFP portion does as well. I don't have time to go into all the data, but that's just one example. So consistent with the idea that Caesar-1 is protecting, we see a correlation and there's an adoption of small RNAs in the Caesar-1 pathway targeting the GFP. And then, of course, now she has an assay. Me too has an assay for doing this, for looking at RNA activation. So she can cross these two strains together and she can see the nuclear GFP-CDK signal come up. And so she did a very simple experiment. She made a Caesar-1 het, and she crossed it. She was going to make the homozygote, but she's lucky, and the experiment worked even with a het. Even being heterozygous for Caesar-1 in the mother or the father is sufficient to prevent that transactivation phenomenon. So the first generation cross progeny failed to turn on the nuclear signals, completely abolished when you're only heterozygous for Caesar-1. So it's very dose sensitive. And again, consistent with the idea that Caesar-1 is indeed functioning like a memory of previous gene expression. And since I'm running short on time, I'll just sort of like reiterate what this seems to suggest. The animal is using its Argonauts to remember what genes were active in the germline in the last generation. And by doing that, it's able to use its piRNAs to scan for and recognize foreign sequences and then initiate silencing that's permanent on those targets. And it's an amazing system. And I'll just take you through the model quickly. We think, and we haven't shown this, but we think that Caesar-1 can recruit RDRP to an adjacent sequence, and for some reason it prefers maybe to do it this way rather than that way. So I've shown it here on a transgene where GFP is tagged at the 3', and you can see that if this happens first, then you'll develop protection against the PIWI pathway. And if this happens first, if PIWI recognizes GFP first, then you'll get RDRP recruited and you'll load the WAGO pathway. So you have these two sort of opposite outcomes that are possible. Once this is recognized by Caesar-1, Caesar-1 can prevent PRG1 from being recruited there. If you cross them together, if this is a weak protective signal, this can win and turn that one off, and then it could stay off. If this is a strong protective signal, or if it accumulates long enough over time, then it can actually turn this one on. So these are like dueling pathways, and it's really fascinating biology. And all of this is being played out also at the chromatin level, and we're just beginning to probe how the chromatin changes as alleles change from an active to a licensed to a silent state. And there's just a whole lot of work still to do. And so now I'll just end with one more picture. This is a picture of my daughter Victoria in Stockholm at the Nobel Prize headquarters, which is an amazing place if you ever get to go there. They're absolutely full of gold medals. They're everywhere. And so I just told Vicky, you know, grab as many of those as you can. And so she's grabbing them. And it's kind of a poignant slide too, because Vicky's been type 1 diabetic since she was 1.5, and each one of those gold medals has chocolate inside. And for her to eat that, she needs to have insulin, which she's got right here. She's got a pump on, you can't see it, but she needs insulin every day or she'll die. And every child like her until the 20s died of type 1 diabetes. It was a death sentence. And it was a dramatic, dramatic triumph of medicine, maybe the first real triumph of medicine ever, you know, when you think about it. What Banting and Best and McLeod, their group did, Toronto, just in the course of a couple of years, transformed the history of this one disease. And you don't appreciate how important the work all of you guys are doing until someone you love is alive because of modern medicine. The thing that, again, is amazing about this is that bacteria read the human genetic code. Think about all that time back there. The innovations, the real innovations in the history of life occurred way back there when we were common ancestor. Our common ancestor was with bacteria. Bacteria can still read the human genetic code and make Vicky human insulin that keeps her alive every day. I think it's an amazing story of a triumph of medicine. And it's the kind of thing that we need to make happen more and more frequently, and I think we will. I mean, we are on the verge of a really breathtaking new era in the history of molecular biology and medicine, and I think that there's a huge amount of work to do to transform the lives of many other people. And I thank you all for your attention and encourage you to go back and have fun in the lab. And I won't go through the acknowledgments right now. We are hiring. That brand new building, you know, assuming sooner or later there'll be funding. So please send your applications. And I'll stop there. I'd be happy to take questions.
Maybe we could just start with talking about translational research, you mentioned diabetes here at the end. In your mind, what is going to be the biggest impact from your discoveries to translational research in medicine in your lifetime or beyond?
I think, to me, it's clear that despite all the hype, I mean, I totally believe in the idea that RNAi can be a drug in its own right. I think clearly the way that RNAi is really working for us is in the laboratory, where it's allowing us to put genes into pathways. You know, as we discover new human genes involved in disease, it's allowing us much more rapidly to understand where that gene fits into a gene expression program or a signaling pathway. And I think that that's already having an impact. It's actually surprisingly hard to trace the impacts of your research on things like new drugs. I mean, there's occasions when there are things like the Banting and Best insulin discovery that lead directly to a new drug. But a lot of the things that, you know, like CRISPR or RNAi, it's going to take years and years before those technologies become a drug. And I think we need to do a better job as a community of accounting for the payoff so that we can, you know, get, you know, our fellow citizens in Congress to buy in on the value of funding research. So I think it's actually really, you've raised a really good question. I think the major impact is in the laboratory, and I think it's actually having a big impact, but I find it kind of hard to, you know, trace where that impact is and how long it will be before it's really clear in terms of a drug pipeline affecting people. Anyone?
Well, you all have questions? Someone? Anyone? Sure. Right here. Can we get a microphone?
Hey, so you sort of mentioned the CRISPR system. I just want to hear your thoughts about why we don't have a CRISPR system. You know, the bacteria, prokaryotes have the CRISPR system, and why don't we have the CRISPR system? Or the other way around, why some of the bacterial Argonaut-like hogs we don't really know what they're doing in the context of these bacteria?
Yeah, that's a great question. You know, and I don't think I can really answer it. First of all, I would say I wouldn't be sure that we don't have a CRISPR-like system. I think that for bacteria are the germline, and the germline has to walk a sort of a tightrope, right? Because it wants to be able to evolve. It wants to be able to have changes that lead to new things. I mean, I'm anthropomorphizing, obviously, but for a germline to be able to evolve, it needs to be able to change. But on the other hand, it's probably very good not to allow new things to get expressed in the germline if they weren't expressed before. So the animal really is walking a balancing act, is doing a balancing act here. I think that we have a lot to learn from studying germline, which is really hard to do in the human or the mouse or almost any animal. We have a lot to learn about how germlines are defended. And keep in mind, the germline doesn't have to be perfect in its defense against viruses. It only has to be better than the soma, because the viruses can replicate in your soma. They don't necessarily need to get into your germline to be able to do their stuff. I think if they needed to, they would. They do occasionally. I think the whole biology, you've raised a question that I can't answer. I think it's likely that there are some really interesting things still to be discovered in that would be an answer to your question.
So I'm sure there are a number of young scientists and physician scientists in training in this room right now. So I was wondering if you had any advice for the next generation that's going to be taking over this work and this endeavor.
Well, I'm not quitting yet. No, hopefully there's plenty of room for all of you to take over. This is really, you know, it's hard to give advice. All I can say is, you know, in my experience, there's two things that are good, and you have to do both. One is you have to be persistent, but because persistence, if you're wrong, is bad, which is most of the time, you also need to constantly be coming up with new ideas. So one of the things that you should definitely be doing is bouncing your ideas around, both with your colleagues and with yourself and with your potential competitors. And every day, you really should be thinking, what else can I do today? Like Masaki's experiment, pulling something out of the trash can. I mean, there's things like that that I've seen it over and over again that people ignore and ignore and ignore. It's a little piece of weird information that you don't understand, and you ignore it and ignore it and ignore it. And if you just would act on that, just do something, do one experiment every day. If it doesn't take too much time, what the heck? Start a new experiment. That's why if you combine that with persistence, it really pays off, because persistence is good to a point, but it won't start new projects for you. You have to start them on your own. And so I would just urge you all, also don't tell your PI. Just do it. Tell them later. They love, love, love to be surprised that you thought of an experiment on your own. No, you should tell your PI too, but if they constantly say no, then don't tell them. Just do it if you can.
So I really loved your work with the Caesar-1 stuff, especially towards the end. I think it's fascinating. And I don't mean to quote reviewer 3 that you mentioned previously, but do you think, where can the worm be storing all these thousands of memory sequences that it might be using in the future? Is there some sort of like common sort of epigenetic stuff that we already know about, or do you think must it be something else?
That's a great question. Did you review my paper? We just had we have a paper coming out on that stuff. That one is in press at Cell, and the other one relating to more Caesar-1 didn't get into Cell. One of the reviewers said it was more appropriate for Science or Nature, which is kind of funny. It was very flattering, but to see that in a rejection was really funny. And admittedly, we didn't have that key mechanistic insight that would be great to have. So I don't really know how it's working, but I can tell you that the Caesar-1 small RNAs are made by RDRP by using the mRNA, the mature mRNA, as a template. So what we think is that RDRP is recruited to mRNAs and without destroying them is making antisense small RNAs that get loaded onto Caesar-1. And presumably there's some way the organism can tell that it's a previously expressed RNA, and once it's established, this is feed forward and it's maintained forever. And it's not so bizarre if you think about this amazing work from Oxytricha and Tetrahymena, where the macronuclear genomes the actual germline genome is totally scrambled. The introns, the exons for actin can be in either strand, and they get rearranged when the macronucleus is made using RNA guides to rearrange the DNA. And the thinking is, again, that this memory of previous gene expression is used as a template for reformatting, and that's been experimentally tested in these ciliated protozoans. I think that, again, organisms are really, really sophisticated in what they can do. I think sort of the other thing that's strange about the Caesar-1 pathway is it's an incredibly conservative way of running a germline, because you're somehow keeping track of everything. And the paper that did get accepted shows that all the spermatogenesis related gene expression is being monitored by Caesar-1, and the sperm is getting loaded with Caesar small RNAs, and it's like a memory of previous paternal gene expression. And if you don't have that, which we can do because Caesar-1 males are fertile, they gradually become sterile. Within 5 generations, they become completely sterile. So it's just really an interesting biology that they're actually keeping track not only of all the genes expressed in both germlines, but the genes that are only expressed in the male, the genes that are only expressed in the female. I think one of the really thing that's important to remember about this is worms are doing this. So I gave a talk to the Congressional Biomedical Caucus, and I said, surely if a worm can do it, humans must be able to do something like this. But the point is, you study a model organism because you want to learn about what's possible, right? It doesn't necessarily mean that's how it works in the human. But I wouldn't be surprised if in the germline anyway, the human genome is managed in a similarly conservative way.
I have a question regarding your ego concept that they remember. So how about one microRNA can target one messenger RNA in one organ and different target messenger RNA in different organ? If that messenger RNA or piRNA can remember what they should bind at the RISC complex, why it's different in different organs?
Are you talking about microRNAs? Well, the microRNA system may be very different from the one that I've described here, although I think it would be really interesting if it turns out that all cells, to some extent, are keeping track of gene expression programs. Think about it. We all know about it. We often call it epigenetic regulation when we mean chromatin-mediated regulation. There's a lot of regulation at the level of the gene that occurs. You can activate a gene, and it can be transcribing, and it can be making RNA that is expressed or is subject to silencing. Even silent genes are being expressed. The transcripts are being fed into the silencing machinery instead of the protein synthetic machinery. So I think that it's possible that, I'm not sure this is what you're driving at, but it's possible that microRNAs are actually able to ignore targets of some RNAs that are meant to be expressed in a particular tissue, and that there's some licensing factor that's present on certain mRNAs. You could also change the 3' UTR. The part that I'm alluding to here is a science fiction idea that microRNAs might actually be able to discriminate between a transcript that's so-called licensed for expression and a transcript that is not licensed for expression in that tissue. And that's totally fanciful, but I don't see why it wouldn't be possible for there to be some leaky transcription that's occurring at a gene that shouldn't be on in a certain cell type, and microRNAs are cleaning that mess up, making sure that they don't mess up the cell. It's possible microRNAs are doing that and we don't appreciate it yet. We're focused more on some of the big things that they do where they're central in the regulation of a particular transcript. They might be doing a whole lot of housekeeping that we're not aware of because we don't know how to look for it. I don't know if I really answered your question, but I think that was sort of the general gist of what you were asking.
So Eric Kandel's group has shown that piRNAs have a role in learning and memory in the sea slug. And so I was just curious if you think that piRNAs also have roles in differentiated somatic cells that could also be just as important as their role in germ.
Yeah, that would be fascinating if it were true. I read the paper from the Kandel group, and while it was provocative, I didn't find it, the way that it seemed, if I remember correctly, to be working was by regulating the expression of a gene involved in synaptic transmission or something. It wasn't, what would really be cool is if the memory itself was somehow stored in a piRNA, binary code or something like that. I mean, that would be like total sci-fi and really, really cool. And that was sort of what I was hoping the paper would say when I started reading it. But I wouldn't rule out something like that. I just don't see how it would work in the nervous system. But I do think that there's a huge role for post-transcriptional regulation in the nervous system because you have all of these important cellular decisions that need to be made very far away from the cell nucleus. So you need to have a way of regulating gene expression at the synapse. And one great way to do that is to manage translation at the synapse, for example. So that's a great way that it could be involved. But as far as actually, in this case, the small RNAs are actually storing information that's being transmitted, and it's information that's very gene-specific, for example. Could something like that work in the nervous system? It'd be fascinating if it does, but I don't think the Kandel group has shown that yet. So. Well, I'll tell you, one of the most fun things about the Nobel Prize is you get to tell your neighbor what you work on, and they actually listen to you. They think all of a sudden that it must be important. So that's a lot of fun. But it's funny that like 2 days after the prize, one of my neighbors was dragging their trash cans out, and I was dragging mine out from the other side. And they came up to me, and we met in the middle of the street, and she said, this is like 3 days after the prize, she said, some things never change. And they did listen for a little while, but now they don't even know, or so it's fun. It's a lot of fun. The Nobel Prize shines a spotlight on science in a way that no other recognition does, and I think it's wonderful that happens every year, and that it's so, it really, just for that 1 week in October and then another week in December, it really shines a light on science. And I think more and more, people appreciate science, and they appreciate it when they're sick or something's not working, but I think more and more our society really depends on science, and we need to do a better job communicating that all the time. And that's why I'm so happy to be here with the Nobel Prize group doing this, to try to continue that and to encourage all of you guys to make that effort, as hard as it is to tell your neighbor what you work on and why it's important. I find the audience is very receptive to medical research in particular, and they really get things like, bacteria, even though it's genetically modified bacteria, it's bacteria doing something really good. They appreciate that it makes insulin. So I think one of the great things is that it does give you a voice, but it also reminds you that you need to multiply that, because you can't do it yourself. You need everybody to get out there. There's really important elections coming up in 2014. Anyone who wants to be funded in their future probably should make sure they vote. But certainly there's a lot at stake, and I think that's one of the great things about it, is that opportunity.
Last question.
I want to ask you a little bit about competition in science, because obviously it has many pluses in motivating, but there are distinct disadvantages, and maybe as a society a lot of things could progress differently. So how do you see science evolving and maybe decreasing competition, increasing more collaboration? How can we all move in that direction?
That's a great question. I don't have an answer to that. What I can tell you from my personal experience, that even under the most highly charged situation that I've experienced, where I felt very threatened by a competitor, in the end, talking about it with that competitor turned out to be absolutely the best thing I could possibly do. And going directly to them, making a phone call, sending an email, reaching out to your competitor. Somebody, I don't know who it was, said, nobody in the world cares more about your work than your competitors, right? So, and if you think you're about to scoop a competitor, that you're going to blow them out of the water, right? You probably should, long before publication day, let them know that you're on this, you've got this breakthrough result, and you should let them know. It's not a game we're all playing here. This is important stuff. People's lives are literally at stake, and the faster things get discovered, the better. Now, I'm not saying you should collaborate with them necessarily, but you should at least let them know. And 9 times out of 10, they have something that's really interesting that could speed you up a lot. They have a tool that you could use. They have an insight that you haven't made yet. Maybe they're not ready to submit their paper, but they've got a really great story cooking, and it's a piece of the puzzle that you don't have. You talk to them, you may not collaborate on the paper you're about to publish. The next paper might be the best paper you've ever published, and it's going to be because you're collaborating with that person now. That Andy and I were like that. We were working in parallel, keeping each other abreast of what's going on. We came together to work together around the double-stranded RNA as a trigger and the whole RNAi phenomenon. It was just really fascinating, fun science, and I encourage you all to do that. Don't try to scoop your competitors. Definitely don't. You know, if they do it to you, so what? You know, find somebody else to collaborate with maybe if they're not being collaborative. But yeah, it multiplies what you can do. And it's usually the person you don't know about who's going to scoop you anyway. So you're much better off if you learn something that speeds you up, because you're going to get scooped by someone you don't even know is working on what you're working on. So I tell people that all the time, and it's amazing how different people respond to that. I know it took me a long time to feel open about sharing what we're working on, and people in my lab get very nervous sometimes when I talk about what they're doing. But I really encourage you to do that. I think you'll find, in fact, that you'll get scooped less often if you do that. So that would be what I would say. It's a great question.
We just would like to again thank MedImmune and Nobel Media, and especially Dr. Mello for being here today.
Thank you. Thank you.